Yeah, so as Susan said, I'm Curtis Carlson. I'm a PhD student at UC Davis. I'm here also with Dr. Christine Tabuloc, a postdoc in our lab. We work in the lab of Joanna Chu. We do circadian biology. And I just want to thank everybody for inviting us here again. We were here 2 years ago. It was a really eye-opening experience. Great to see familiar faces, meet new people, and really a great chance to update you all on the things that we've been doing, as well as introduce the new faces here to some of the work that we've been doing, where we're using fruit flies, those little flies that are flying over your bananas in your kitchen, how we're using those to not only understand the basic biology underlying SKS to really figure out what's going on, but hopefully to use these flies to, as Matt alluded to, find potential drugs that we can repurpose and get to your kids in a much quicker fashion. So as many of you are aware, as we just heard too, there's no FDA-approved treatments for SKS, and really very little is actually known about the underlying pathophysiology of SKS. And so our overall goal is to actually develop fruit fly models of SKS so we can understand the molecular mechanisms that are leading to some of these symptoms, and then ultimately use these in a drug repurposing screen. So we have a pipeline of how we're doing this, where we first generate fly models, where we actually introduce SKS alleles, SKS variants into the flies, either through CRISPR or other transgenic techniques. We then take those flies and actually characterize some of their behaviors to see, do they mimic some of the symptoms that SKS patients have? Once we can see that those flies indeed are showing similar symptoms to SKS patients, we can then import them into a drug screening platform where we can test some of those FDA-approved compounds to try to see, do any of those alleviate some of the symptoms that the flies are having. And then those drug screenings actually also inform some of the behaviors. So if we get drugs that are working, we can move them back into other more complex behaviors in the flies to see if they alleviate some of the other symptoms. While at the same time, we're also doing this molecular characterization, as I said, where we're really trying to figure out, look under the hood, see what are these mutations doing to the animal, to the cells, to the whole body. And then again, doing that kind of work actually really informs our drug screening, where once we can figure out some of these pathways that are being perturbed, then we can actually look at more, maybe a more narrower candidates of drugs and test those out. And then again, just this as science is a really iterative process. So finding drugs, going back to the molecular work, going back to behavior, it's a back and forth kind of game. So some of you might actually be wondering, what does a fly have to do with my child? So I'm here to try to convince you that flies are a very powerful model organism. So what makes them valuable for this kind of work? So flies have a very rapid generation time. They go from egg to adult in about 2 weeks, which makes them a very economical and high throughput model system. So they're pretty cheap to work with. And flies have the great benefit of well over 100 years of genetic studies using them. So there's a really diverse array of genetic tools available to work with in fruit flies. So we're able to manipulate the fly's genetics in virtually any way that we want and study the outcomes. And really importantly, despite being an insect with 6 legs and wings, they actually have analogous anatomy to humans. So inside the fly, they have organs that are doing the same things that's going on in our body. And really importantly too, flies have already a very established use in circadian and sleep research, and flies actually sleep. Flies indeed also sleep at night. And probably most importantly to this community and this project is that the role of mTOR in development and metabolism is conserved between the drosophila or fruit flies and humans. So in the fly, mTOR is doing the same thing that mTOR is doing in our own bodies. And remarkably, so this list keeps growing of SKS variants that are being detected and reported. But of the at least 52 that I have in my list, 42 of those occur at conserved residues between flies and humans. So in the fly's mTOR, that same variant that your child might have is actually conserved in the fly. So we can actually go in and change their genetics to then mimic the patient allele, patient variant. And all of this The great benefit of the flies, as I said, is the fly is a very rapid platform to do a lot of this work, where we can pretty quickly generate these models, fairly quickly characterize their behaviors depending on what we're looking at, do a lot of molecular characterization pretty rapidly, and then again, you can actually do some of these high throughput drug screenings in these flies. And this is not to discount mammalian models or mouse models. Those are extremely important models. But the point with the flies is that we can get to the mouse model much quicker with more informed strategies. So we can find things that might work in the fly, and we can bring those into the mouse or human cells or even patients or something like that and test them in that. So it's a way to kind of speed up the science. And what we're doing here really is nothing new. We're not really blazing new ground. Flies have been used to model rare diseases and human diseases previously, so it's really well known. And there's a real challenge with studying rare diseases and getting therapeutics or anything because of the time and cost it takes to develop a lot of these vertebrate models, such as mice, rats, non-human primates, that kind of thing. And the fly offers a really robust and rapid system to study diseases. And remarkably, in the fly's genome and its genetics, nearly 75% of disease-related genes in humans are actually conserved in the fly. So people have used the fly to study all types of diseases in humans, such as neurodegeneration, muscle degeneration, intestinal dysfunction, diabetes, all sorts of things. So the fly really is a great model for these kind of systems. And as you all are aware, SKS is complicated, and it's complicated because mTOR is an extremely complicated, I don't like to call it a pathway, I like to call it a network. And really mTOR touches nearly everything in the cell, everything in our bodies. So mTOR regulates brain activity and seizures, it regulates growth and metabolism, and it regulates sleep and circadian rhythms at different levels. So what we're trying to do with the fly really is study at all of these levels to try to really understand what is going on in this rare disease and how do these different sort of modules interact with each other, and how, when we perturb mTOR, how is it disturbing all of these? So really studying it from multiple angles. So the way that we have done this right now is we've generated many different SKS alleles in the flies. This is not an exhaustive list. Again, every time I get updates from Susan or other people or check the literature, there's more and more variants popping up. But what we've been trying to do right now, at least, is find some of the most common variants among the population and find if they're conserved between flies and humans, and generate those ones, looking at different domains as well, hoping to cast the widest net to see if we can make the biggest impact for the most people, and see how do these mutations in different domains lead to different patient outcomes, different symptoms. And so we've done this by making germline SKS flies. So these are CRISPR-edited flies, where now they carry every cell in their body actually carries the SKS variant. And those are shown here in red on this diagram. And then we also have another flavor of fly here where we can actually sort of simulate mosaic SKS. I know that's a very big deal, where we can actually use different genetic techniques in flies to target the expression of these SKS variants to different tissues, so we can understand what is going on locally, like if we just express this only in the fly's brain or only in the fly's muscles, that kind of thing, to study the outcome there. And those are shown here in blue. And many of these, we actually have both versions as well. So we have the germline and then the sort of mosaic flies, so we can kind of tease them apart. So since we met last time 2 years ago, we've actually generated 4 new germline SKS flies, again using CRISPR, where you can actually edit the fly embryo, then the fly inherits the SKS allele, grows up into an adult, and then we can use those flies in our experiments. So we've generated two germline flies with variants in the fat domain, as well as 2 in the kinase domain that we're working on characterizing. And remarkably, and this really blew my mind when I first started doing this work and saw this, is that when you introduce these mTOR variants into the flies also get sick. And they get sick in very similar ways to some of your kids. So we see that the flies actually phenocopy or mimic patient symptoms in a variety of ways, where they have sleeping symptoms. So here is a little sleep profile. I'm going to show you first. Right here is plotting just what a normal healthy fly sleeps like, where they have daytime siestas. They wake up kind of in the morning and wake up in the evening. Whereas here we have a SKS fly that is sleeping much less than the control fly. And I'll get into it a little bit, that also the quality of this sleep we're looking at. So it's not just the amount of sleep, but also the quality of that sleep. Our flies also exhibit seizure-like behavior. Well, maybe this video won't load, but point being that when you introduce some of these mutations into the flies also can have seizures. So we're really looking into that now as well. As many of you know, your kids tend to be hyperphagic. They overeat. And indeed, when we do the same thing with our SKS flies, put them into this little assay where you give them a measured amount of food, you can see that SKS flies also consume more food on average compared to control flies. And as we've heard too, that patients tend to have body temperature dysregulation or interoception problems, or they can't quite sense the outside world properly. Indeed, our flies also seem to be having trouble sensing temperature as well, or they tend to be running kind of hot, and so they tend to prefer to cool themselves down and hang out in cool sides of arenas that I'll mention in a little bit. So for today, for the sake of time, I'm only going to focus on showing you a little bit of sleep symptoms and a little bit of that body temperature dysregulation before we also get into the drug repurposing with Christine. So again, when we have these germline SKS flies, they exhibit sleep loss symptoms, where when we look at the total amount of sleep that those flies are having, you see here in red, we have one of the SKS variant flies. They have significantly less amount of total sleep over the 24-hour day. But again, more than just total sleep, we want to look at the architecture of that sleep. You can sleep a lot, but if you're waking up every 5 minutes, you're having the micro naps, you're not getting that full regenerative kind of sleep. So we can see that on average, this variant, these flies have fewer numbers of sleep bouts. So they're going to sleep less often, but the length of those bouts is actually more or less about the same as controls. So they're really just not getting a lot of sleep. Then when we look at other SKS variants, just as we all know here that one child has very different symptoms than another child. So when we look at these other variants, we see not only does this variant have less total sleep, they have about the same number of sleep bouts as a control fly, but significantly shorter bouts of sleep. So in other words, this fly has very, very fragmented sleep, never really gets into the deep homeostatic recovery, regenerative kind of sleep that we need. And again, another variant here, less total sleep, fewer bouts, but actually about the same bout length. And so what we're seeing too, even amongst our flies, is a vast spectrum of symptoms, very, very similar to what the patients are having. Last time we were here, we heard a lot of anecdotal stories of how their kids can go out into the snow with no clothes on. They don't recognize how cold it is. They'll do the kind of opposite behavior in heat. And then we have started to hear that some families were finding that if they actually cooled the room down, that their children tend to sleep a little bit better throughout the night. So we decided to test this in our flies to see, do the flies also have similar kind of behavior? So we put these flies into this temperature preference assay. Flies are cold-blooded, so they can't regulate their internal body temperature the same way we do. They regulate that by positioning themselves in the environment. So they'll go, if it's really hot out, they'll go find a cool place to hang out. So when you put these flies, these SKS flies into this arena, you see that SKS flies actually tend to hang out in the cold side of the arena. So that implies that they actually tend to be running hot or something like that. So they're seeking the cooler side of to really regulate their internal body temperature. So we saw this and thought, what if we just put these flies into like colder incubators and measure their sleep? And indeed, when we took these flies that we normally do these assays at 25 degrees Celsius, moved them into 21 degrees, which seemed to be about what they were preferring in this assay, it indeed improves their sleep, where the SKS fly now in the colder temperature is actually sleeping about the same as a wild type regular control fly at 25 degrees. And really, when you break down this sleep too, it's actually getting even more consolidated sleep than compared to the control. So while both controls also improve sleep in cold, the SKS fly is actually gaining more benefit from it than even a control fly. And then because one of mTOR's biggest jobs, its main thing that it's really doing is it's a nutrient sensor in our cells where it's looking for growth factors, amino acids, energy like glucose, insulin, that kind of thing. And that's what really is turning it on and turning it off to regulate cell growth, like telling your body to grow and that kind of thing. So because we saw this, we thought, maybe diet has an impact on some of these sleep symptoms or fly symptoms that they're showing. So we took control flies and SKS flies and actually fed them varying degrees of nutrients, from low nutrients to high nutrients. And in wild type healthy flies, it's really well known that the better the food you give them, the more they sleep. Just like us, you eat a big meal, you want to go to sleep, you'll have better sleep. Whereas with the SKS flies, unless you go to the extreme end of very little nutrition, they don't gain any of that benefit from additional nutrient input into them. So they kind of plateau. So this sort of implies that these mutations might be affecting the way that mTOR is actually sensing nutrients, that it's not as responsive as a healthy copy of mTOR. And so I think this really is kind of highlighting that we really should possibly be thinking about how is our diet, how is our children's nutrition really impacting some of the symptoms that they're having. And I personally think that that should be a really good conversation you should have with your clinicians. Maybe get a dietitian or something like that to really start monitoring what kind of food your child is having, because maybe more nutrition is not actually doing anything for them, or you might need to have more tailored diets that could potentially alleviate some symptoms. But it's implying that mTOR, in this case, can't sense nutrients the right way. And I just want to pause here real quick from the science, or from that sort of fly biology and everything, to really highlight some of these research milestones. We heard a lot about it yesterday, and this is not an exhaustive list at all, but I think it's really remarkable that just about 10 years ago, or 13 years ago now, we had the first description of SKS from Dr. Smith. And since then, we've had a large accumulation of case reports and publications in the literature. We've had, actually probably right before this, we've had the first SKS cell models from Dr. Andrew Liu. We've had the first SKS fly models appearing here in 2022. Andrew got the first NIH grant to study SKS, developed the first SKS mouse models to really study this. We've developed the first CRISPR SKS fly models. And as Susan said, we got the first NIH fellowship to study SKS. And I think all of this is really important. I know sometimes you hear scientists talk about publications. Thank you. I know sometimes you hear scientists talk about publications, searching for journals to submit things to, and you wonder, what does that have to do? I need answers now. But really, all of this is really important because it really sparks interest in the broader research community. The more papers that are out there about this, the more we talk to other scientists, the more the NIH starts to recognize this. It's really showing that there's a lot of interest and a lot of drive to really push SKS research forward. And then really briefly here, I won't go into exhaustive detail, in collaboration with Andrew Liu, we're really trying to dig deep under the hood to figure out how is SKS different from TSC. They're both affecting the mTOR pathway or mTOR network, but clearly treatments in TSC don't work exactly the same way they do in SKS. So we're really trying to look under the hood and get really detailed look into what's going on. So we did some cell modeling and proteomics to really look at what's going on, where the big difference between TSC and SKS in terms of mechanisms is that in TSC, you're kind of losing the upstream brake on mTOR, telling it to stop. Whereas, as we all know, with SKS, it's affecting mTOR itself. And when we did this sort of experiment, looking at the results, we see that there's very, very little overlap, at least in these cell models, between TSC and SKS, where they kind of just share a lot of mitochondrial upregulation, so upregulating energy production. Whereas we see a lot of things very unique to SKS, where it appears to be really rewiring a lot of networks in the cell. And again, as I've seen in the flies, we see kind of in the cells that they really have a reduced response to nutritional input. So when you add nutrients into the cells, very little is actually changing. They're not really sensing the nutrients the way that a healthy cell would. And this kind of work is really, really important to really figure out and start to understand how is SKS perturbing cellular pathways. And through this work, we can actually get more informed and targeted strategies that we can find pathways that are affected. We can find drugs that we know affect those pathways, and then we can take those drugs and introduce them into the flies and see if those drugs do anything to alleviate the fly symptoms. And then again, science is an iterative process. So once we see results in the fly, we can go back to the basic biology. And so with that, I'm going to introduce Dr. Christine Tabuloc, who's going to tell you a little bit more about how we're now using these flies to do drug repurposing screens.
Okay. Hi, everyone. Thank you so much for having us here. Okay, I will keep an eye for time. Okay, so as Curtis mentioned, we are trying to develop this high-throughput drug repurposing screen using our SKS fly models. And so there are several things that we had to consider in order to have an efficient drug repurposing screen. So we first needed to identify a behavior that phenocopies or is related to patient symptoms. So what is a behavior that we see in our flies that we see in human patients? Or is there a behavior that's related to symptoms that we see? We also want something that is high throughput, meaning that we can screen many drugs at one time. One time meaning like in one sitting, for what we can do in a fly model. So of course, if we had robots, it would be a much higher number, but what we can do feasibly with our system. We also want something that has a very clear, distinct difference between our control, so a fly without SKS, and our SKS model. And so what we chose to go with is motor deficiency. So flies have this natural propensity to actually climb up when they are present in a tube. So what we do is we take a tube of flies, we tap that tube down, all the flies will collect at the bottom, and then they naturally just want to climb up. And we want to see how many flies are able to climb to the top in a given time range. And so the reason we chose motor deficiency is because we do know that some patients have motor deficits. But on top of that, this particular assay could also induce seizures when you tap the flies down. They also have an inability to hold on to the vial sometimes. Also, it is related to their activity level. So we know that there are sleep disruptions. So it's a very good behavior that it is like really quick to screen, but is related to a lot of the things that are high on the priority list in terms of symptom management. So as I mentioned, we wanted to do something that was high throughput in our particular system. So we can actually screen 20 compounds in 2 weeks. So in a 2-week period, I can go through 20 and then get some results from that. And in terms of the clear difference, so here I have 2 vials. I have the control flies on the left, so they don't have SKS and our SKS models. You could see that most of the flies are actually climbing to the top in the control, whereas most of the flies in the SKS model are actually hanging out in the middle and the bottom of the vial. So this enables us to do video recordings and then really quickly see whether or not a drug is going to make a difference in this particular phenotype. The other great thing about this particular screen is that it's actually a very well-established drug repurposing screen in fly models. So if you were here a couple of years ago, we had Dr. Clement Chow show some of his work, and he's actually done a few drug repurposing screens using this particular system. So with the help of Matt and Kat, we were provided a pilot screen of 20 drugs, all that are specifically targeting mTOR or the mTOR-related pathways, so they are inhibitors. And the goal of this pilot screen really was to see what is the feasibility of this assay. Can it be done with the amount of drugs that we want to screen? Is this really truly a good phenotype for what we want to look at? And then use this information to determine whether we need to make any changes on our end to actually make this drug repurposing screen work. So the first thing that we did is we started with mTOR pathway-related inhibitors, and then we started with one variant. And so we decided to go with the C1483F variant, and this is because it is the one with the most amount of patients that we can model in our fly system. That's not to say that we won't screen others, because we will, but this is just a starting point. So what this looks like in principle is we were given 20 drugs that I did not know the identity of, so there's no bias here, it's completely blinded. So I listed them just as their position on the plate that I was given. I have 24 vials of flies. So we have 2 of our control flies, 2 of our SKS model without the drugs, and then we have the rest all with the drug. And what that looks like in reality is something like this. So I'm recording videos based off of that system there. So the next question then becomes, now that you have this platform, how exactly are you going to be giving the drugs to the flies? So we decided to administer the drugs orally. So we actually mix the drug into the food, and then we give it to them in 2 different ways. So this is what I'm calling a chronic treatment versus an acute treatment. So in the chronic treatment, this means that we are giving the flies the drug to the parents, so all the way from the embryo, like egg stage, all the way throughout their adulthood, which is the most common way of carrying out a drug screen. However, this is not really reflective of what we're going to see with patients, right? So we're not going to be giving them the drug prior to them being born. And so what we decided to do was, in addition to doing it this way, is to actually do what we're calling an acute treatment. Let's feed them the drug just during the adult stage, and then see whether or not there's going to be a change in symptoms. So because we don't know how long it will take for a drug to have an effect, we ran the assay for several days. So I did 7 days post-treatment to see whether or not there would be an improvement in this climbing ability. So of course, we want to be able to make the most robust conclusion. So we built in a lot of different things here, so that way we're very confident in what we're saying about these hits when it comes to the actual drug screen. So we actually use 3 groups of flies per compound per treatment type. So what that means basically is that little rack of flies I showed you guys, I have 3 of those for every assay every day that I'm recording. And then each of these flies were actually subjected to the assay 3 times. So tap down, let them climb up, tap down again, let them climb up. And then we can take an average of the amount of times that they do that, just so that way we're getting the strongest and clearest data. And then finally, we recorded over multiple days to see whether the phenotype persists and to make sure that we're not having too many false negatives or false positives if we're only just picking a snapshot of 1 day. So when we do the real drug screen, we were going to be calling these hits by assessing the changes in distribution of the flies throughout the vial. So on the y-axis here, I have the number of flies that are climbing to the top. And on the x-axis, I have the different treatments or genotypes. So the first bar here is our control flies. So majority of the flies are going to make it to the top, which is that white bar there. And then in our untreated SKS flies, we have majority of the flies being more towards the middle or the bottom of the vial. And so the dotted red lines here, I guess they're not dotted, they actually just show where the top, middle, and bottom are on the untreated SKS fly. So I'm comparing everything to that particular bar. And what this will look like in principle is I'll have a bunch of different bars for all the different treatments, and I can see whether or not we see an improvement in the number of flies going from bottom to middle or middle to top, compared with the untreated SKS flies. So I just wanted to show an example of what the data looks like. So this is just a single group on a single day. And we basically just track that progression. So this is either one of the acute or the chronic. And so what that looks like within a vial is something like this. So again, we have our control flies that don't have SKS, and then our SKS model, where most of the flies are either at the bottom or at the top, middle of the vial. And then when we actually administer one of the drugs that are inhibiting the mTOR pathway, you actually see an improvement of climbing, where majority of the ones that were at the bottom are now in the middle or the top. So now that we've kind of looked at the feasibility of what this assay looks like, our next step would be to do a much larger scale screen. So basically what we will do is we'll partner with Matt and NCATS to have an FDA-approved drug panel. And with that panel, basically we can get an array of results. We can have some things that are positive results, meaning we see major or minor improvements in climbing. Or we can have a negative result, meaning that either there's no improvement at all, or maybe in some cases it might be worse, right? And then we would have to figure out why that's the case. And so if we are to see a positive result, what our following steps would be is to actually repeat the assay again with the other variants. Is it that it just helps this particular variant, or is this actually gonna hold true for other variants? And then we can also test other phenotypes that might not be as high throughput as this particular screen. So the concern would be, just because it works in climbing, does that mean that it's actually gonna help with seizure? Is it actually gonna help with sleep? Is it gonna help with hyperphagia? So all of the behaviors that Curtis showed you guys, we can just take our positive hits and then run that in all of these different assays. And we're still working on characterizing more behaviors as well. Okay. So further down the line, what we could also do is, based off of which drugs are actually hit, we can identify pathways that are involved in the symptom manifestation. And then that will enable us to identify even more compounds that might be making a difference that we wouldn't have considered to begin with. And we can also use the data that Curtis and Andrew have gathered to help inform our studies as well. And then we could start thinking about, based off of how these drugs work, what are the long-term consequences? What are some of the side effects that we may see? So that's kind of the idea moving forward. And then from there, we can actually start considering these drugs for preclinical or clinical trials. And then, for instance, with the negative results, we can't be confident that it's truly just a negative result. And this is because all the assays that we're doing, because it is a screen, we kind of have to do it quickly. So we're doing one concentration, one dosage. So it could be that maybe the dosage we're using is just toxic. And so if this is a problem, then we would need to do a dose response. Can we try different dosages? And is that actually going to have an effect? Is it truly a negative result, or is it just a factor of the experiment that we are doing? And so we can adjust the dosing for these, and then we can rescreen those particular compounds so we can truly positively, so we can actually discard it and not use it for our purposes. And then if it does improve based on adjusting the dosing, then we would just run it through the positive result pathway here. So that's kind of where we are moving forward. And so with that, I just want to thank our PI, Joanna Chu, as well as all of our previous and past members of the lab who have worked on this project. Of course, our collaborator, Dr. Angelou and Matt and NCATS, for generously providing all the compounds that we need for this. All of our funding sources, for the foundation for being so amazing. And a very, very heartfelt thank you and gratitude to all of you who've been sharing your stories, which helps inform what we do. And for trusting us to do such important work. So just thank you. And we're happy to take questions if there's time.